Hello guys, today's our lecture about cell fractionation, which includes extraction, homogenization, and centrifugation. So if we make a definition for cell fractionation, we say cell fractionation is a procedure for rupturing cell separation and suspension of cell constituents in non-isotonic medium in order to study their structure, chemical composition, and function. Carta cell fractionation أو procedure كتيدا إما cell rupture دكين component كاني ناوي separation دكين و suspension دكين لنوع isotonic medium لبو أوي study structure يمكن يعنيش chemical composition بزانين chemical composition هر component تشي سيلي تيا يعنيش function كانيان هر component تشي سيلي function تيا cell fractionation involves three steps أو عملية step لخوي تقرأتن. The first one is the extraction, the second one is homogenization, and the third one is centrifugation. So let's talk about each steps of the cell fractionation in a detail. The first step is extraction. Extraction is the first step toward isolating any subcellular structure. كم step لبو لك تذكر نوعي هر cellular component تشي extraction. In order to maintain the biological activities of organelles and biomolecules, they may be extracted in a mild conditions called free cell system. And free cell system is an in vitro tool widely used to study the biological reaction that happen within the cell, apart from a full cell system, thus reducing the complex interaction typically found when working in a whole cell. And for this, to maintain the biological activities of each organelles, the cells or tissues are suspended in a solution of appropriate pH. It is 7 or 7.4, I think, and salt content. Usually isotonic sucrose 0.25 mol per liter at 0 to 40 Celsius degree. The condition should be optimal. Should be optimal osmotic pressure, optimal pH, optimal temperature to preserve the function of each organelles and biomolecules. The second step is homogenization. The suspended cells are then disrupted by the process of homogenization. We can say the suspended cells are then disintegrated or breaking apart by the process of the homogenization. And the homogenization is usually done by the grinding, high pressure French press or nitrogen pump, and osmotic shock or sonic. And the last one is sonication, ultrasonic vibration. And in the next slides, we are talking about 4 steps with a figure. And the grinding is done by a pistol or a motor or Potter homogenizer, a high-speed blender. The later constituents of the cylinders are separated by a narrow gap. So, as this is appearing in the picture, it's overall of the 3 steps. The first one, we have a cell suspension, and then we use a mild detergent to make a hole in the plasma membrane. And in the third picture is appearing carefully conducted homogenization leaves most of the membrane enclosed organelles intact. And then we put the homogenate in a tube and cell wash solution, resuspend and centrifuge. And permeabilization, we use a buffer vortex and incubate. Then we cytosolic proteins centrifuges for Silesi, and we collect the next step. We collect the supernatant and solubilization buffer. We use the solubilization buffer and then resuspend and again incubate, and after that we centrifuge at the 4 Celsius, and then after that we collect the supernatant. We obtain membrane proteins. So, as we said before, homogenization is rupturing the plasma membrane and releasing the organelles out. So, here there are 4 common procedures. هربا كنت اشترقت مان homogenization بريتلا rupture كرني plasma membrane release organella كاني ناوخانكا. صار ريغا من هي commonly used لبو rupturing cell بەکاریدێنین. یەکەمیان break cells with a high frequency خانەکان دەشکێنین بە using high frequency sound کە دەنگەکی زۆر گەورە بەکاردێنین لە بۆ ئەوەی کە بتانین پلازما ممبرینەکەی ڕەپچەر بکەین و ئۆرگانێڵەکان ڕیلیز بن. دووەمیشیان using a mild detergent mild detergenتی بەکاردێنم هەزا بتانم هۆڵەکان لە هۆڵی کونی لە پلازما ممبرین پیا بکەم بۆ نوعش خانەکە ڕەپچەر دەبیتن و سێمیشیان بە زەختێکی زۆر لە خانەکان بکەم کە بە هۆڵەکی زۆر گچکەدا تێپەڕ ببن بە زەختێکی زۆر وە دەکاتن خانەکان ڕەپچەر ببنەوە پلازما ممبرین ڕەپچەر ببی و خانەکان break ببن و ببتە هۆی ئەوەی ئۆرگانێڵەکان ریلیز ببن. و چارەمیان shield cells between a close fitting rotating plunger and the thick walls of a glass vessel rotating plungeri بەکاردێنم لەناو thick glassەکی سەڵەکانم دادەنم rotating plunger بەکاردێنم کە هەم دەسوورەتن هەم زەختیشان لێ دەکەوە دەکا خانەکە دیسانەوە لە بۆم ڕەپچەر ببن. بۆ چاڕێی کە commonly used بەکاردێنین لە بۆ ئەوەی خانەکان ڕەپچەر بکەین و ئۆرگانێڵەکان ریلیز بن. So the third and the last step is centrifugation. The separation of the various components of the homogenate is carried out by the series of the centrifugation in an instrument called preparative ultracentrifuge. After we do the homogenization, now all organelles of the cell are together, and this solution is called a homogenate. So in order to separate each organelle, we do the centrifugation, and this is done by through an instrument is called preparative ultracentrifuge. And the ultracentrifuge has a metal rotor containing a cylindrical hole to accommodate centrifuge tubes and a motor that spin the rotor at the high speed to generate the centrifugal forces. So the ultracentrifuge has a metal rotor that holds the centrifuge tubes, and it also has a motor that spins the rotor at a high speed to generate the centrifugal forces, and it also has a vacuum to reduce the friction and prevent the heating and maintain the sample at 0 to 4 Celsius degree. As we said that the sedimentation rate depends on their size and shape of the particles. So different particles, different organelles precipitate at a different rate. This is appeared in this picture. The first tube is contain the cell homogenate, contain all organelles, and it is centrifuged. And after centrifugation, the whole cell nucleus cytosome precipitated. After the whole cell nucleus cytosome removed, that the tube is also centrifuged, and at that time the mitochondrial lysosomes and peroximes are precipitated, and after the precipitate is removed, again centrifuged, and this time microsomes and small vesicles are precipitated. Precipitated is removed and again centrifuged. At the time, the ribosomes, viruses, and large molecules are precipitated. So we can say after each time of the centrifugation, the speed is increased and the size of the particles are decreased. So speed from low to high and the size of the particles from large to small. Marker enzymes for subcellular fractionation, and we have marker enzymes for each cellular organelles. So the marker enzymes are on the cell biomarkers used to characterize a cell type, and they are used in the isolation of the target cellular component. So we have the marker enzymes for each cell organelles. So the marker enzymes for the plasma membrane are 5' nucleotidase and NAK ATPs, and for the nucleus DNA polymerase and RNA polymerase. And for the endoplasmic reticulum, glucose 6 ATPase and cytochrome B5 reductase. And for the Golgi bodies are galactosyl transferase and monosidase. And for the lysosomes are acid phosphate, cathepsin, and beta-glucuronidase. And for the mitochondria, succinate dehydrogenate, cytochrome C oxidase, and oligomycin sensitive ATPase. And for the peroxisomes are catalase, and for the cytosol are LDH and G6PD. So the purposes of the cell fractionation are: the first one is a protein enrichment. Enrich the target protein and improve detection of a low abundance protein. That means repair detection of a low abundance proteins. And the second one is a protein characterization. Identify the subcellular localization of the protein. And the third one is a protein translocation. Monitor translocation of the cell signaling molecules from the cytoplasm to the nucleus. That means it's monitor moving of the cell signaling from the cytoplasm to the nucleus. And today's lecture done. Hope you understand well and get benefits from it.